Good morning. I'm delighted to be in Barcelona at the Cell Meeting on the Med. It's terrific to be back to see so many familiar faces again. So I'd like to update you on developments at Avectas or for people who've been following our development, or for those who are unfamiliar, hopefully to fill you in on our gene editing platform, cell engineering platform, Solupor. So first of all, at a glance, Avectas was founded approximately 10 years ago. It was spun out of the National University of Ireland. We're located just outside of Dublin, where we have now 36 employees. Our Chief Business Officer is based in Cambridge in Massachusetts. We've raised in excess of $50 million in capital to date, and we've an experienced leadership team and scientific advisory board, including luminaries like Bruce Levine, Mark Loudell, and so forth. Each of the photographs here has a particular resonance for me. Some of our shareholders, top left, International Women's Day at Avectas, top right. My colleague Fernando working on one of our platforms, bottom left. And many of you will be familiar with Emily Whitehead, who will be visiting Avectas next week. So our team are very excited about that. So this is our platform, the Solupor platform. It's a platform that has been developed out of market need. When I started this journey, non-viral delivery was against a background of the most embedded means of modifying cells, viral delivery. Non-viral was a sort of an edge scenario, and in the ensuing time it has come much further to the center of the stage, and particularly with the rise of gene editing. And the go-to method for delivering gene editing tools is electroporation, but we're also aware that electroporation can negatively impact on cell cultures, particularly in the face of complex editing and delivering a lot of materials to cells. So our solution, Solupor, sits between these. It's a non-viral, non-electroporation means of delivering materials to cells. It's a physico-chemical method of delivery. We can affect very complex editing and maintain high viability and functionality of the cell cultures. Our platform is embodied in two forms. On the left-hand side is our SUS, or our closed single-use automated system. On the right-hand side, we have an adoption embodiment of the technology, or technology for helping people to adopt the platform, called the research tool. It's open, it's non-GMP, it's more manual. The platforms together cover the span bench to commercial manufacture. And the technology over the last period of time, through some externalization and so forth, has become very robust, consistent, and so forth. The benefits of the Solupor platform, as I've said, we can affect complex editing. The cells which are engineered using the Solupor platform are highly functional. The process is very well tolerated by cells. We see excellent proliferation and viability of cells. We have a defined pathway to GMP, which I'll speak in a little bit more detail about, and the platform is easy to adopt. So to speak for a moment about the pathway to GMP, or our regulatory pathway, first of all, we met during the pandemic with the FDA. There are three aspects to the platform. There's a reagent, there's a controller device, and there's a single-use consumable. Each of these has a documentation suite associated with it, and over the course of the remainder of 2022 and into early 2023, we'll compile the documentation suites and have those available either to file as a DMF or to include in IND owners in the CMC section of their filings. So to speak to some data, first of all, just to baseline everybody, we're showing some single RNP editing in T cells here, showing 3 individual dose responses on familiar targets in T cells, and showing editing between 60 and 80% with very high viability using CRISPR-Cas9. So moving then on to multiplex RNP editing. So in this slide, we're showing a triple edited population of cells, approaching 40% of the population exhibiting the triple negative phenotype, and viability maintained extremely high, near 100% here. So when we do affect these complex edits, the cells retain their high viability. So we then go on to characterize or use analytics on this population of multi-edited cells. And at this point, we introduce a comparator, which is nuclear factor, which is a go-to. It's in most labs. It's a go-to method of electroporating. And what we see is that Solupor is less toxic and results in a higher number of stem central memory cells. So on the left-hand side, we see that the number of early and late apoptotic cells in the culture, which has been electroporated, is greater than in the culture which has been modified with Solupor, or indeed the untreated. And on the right-hand side, we can see that there are a greater number of the type of cells that you want in the culture after Solupor modification rather than or compared to nucleofaction. The apoptosis is at a very early time point after modification, and the phenotyping of the cells is at 4 hours. These are important time points for multi-editing because a lot of the editing happens early. We can also see that the cells proliferate very well after modification, and we can see that the Solupor modified cells are expanding, proliferating better than the nucleoaffected cells. We're currently building on this data by moving into the G-Rex platform. We're showing just some data in the smallest version there, where we're going from about 1 million cells, and we will continue with the G-Rex platform, stepping up to build to a therapeutic dose. And what we see now, and we expect to continue to see, is less debris in the final population and a highly functional therapeutic dose of cells. So in essence, better cells engineered faster. So moving on from that editing to a different model now, which is where we introduced a transient, an mRNA CD19 CAR. We did this not as a candidate therapeutic or anything like that, but just to demonstrate the functionality of the system through killing a cytotoxicity assay. So on the left-hand side, and we'll see on the next slide some animal data, but on the left-hand side, we're showing the RT system, which I showed at the beginning of the presentation. We're showing cytotoxicity of CD19 cells, which have been modified with Solupor and with nuclear factor. On the right-hand side, we're showing our right up to date, our SUS version, which has a very, very similar profile. So when we modified these cells and put them into an animal in this study, on the left-hand side, what we see is at the highest dose at 4 million cells per animal. At day 15, we had on the right-hand side of that green box, we have tumor clearance in 3 out of 5 mice. So we're seeing highly functional cytotoxic cells going in and clearing the tumor within these mice. We published this data in Cytotherapy. On the right-hand side, we can see also that the Solupor modified cells have engrafted to twice the extent that the nuclear-affected cells have in the animals. So we can see highly functional cells. So we've focused our energies as a company of 35 people. We have to, or have had to, focus on our data, and we focused on T cells. However, we're at the stage now where we're expanding that to iPS cells, and I'm just showing a little bit of early data here on delivery to iPS, where we used exactly the same CPPs, the same starting parameters as we've done for T cells, and delivered to this population of iPS. So we see the same type of delivery, we see the same type of the cells seem to be viable after the process, and we're very encouraged by this and are looking forward to deepening this data set by going on to edit iPS cells and so forth. So in terms of 2022, the year ahead, we've raised in excess of $50 million. Our most recent funding closed at the end of Q1. Those funds are to drive the externalization of our technology, which we have been successful at to date and will continue to deepen that, to drive commercial adoption by getting our platforms into the hands of partners who can use them to develop and refine our technology transfer programs and so forth. And of course, the journey to GMP, which is a critical deliverable for the company. And indeed, I look forward to addressing the cell meeting on the MESA group in the autumn, and speaking about how advanced we are at that point with those developments. So I'll be delighted to answer. I'm around for the rest of the day, delighted to answer questions. Our objective is to partner this platform with people who are developing therapeutics and who can benefit from complex editing and cells and resulting cells which are functionally fitter, proliferate better, or differentiate better. So thank you very much indeed.